drop you from the class. Okay, so that's basically what a station would be like, and you would have a minute and a half to answer those questions. We'll go with more like a minute. All right, so those are both what? Formed element questions. So tell me 1A, what would you say? Red blood cell or erythrocyte, and its function was? Carry oxygen. 2A was? Platelet, and its function was? Coagulation or clotting. Bonus. What component in the platelet aids in clot retraction? 41. So 41 to 50 would be bonuses. A lot of times those would be things to see if you can integrate lecture with lab, and sort of with, maybe in lab we're a little anatomy heavy. What would you say for 41? Actinomycin. That's the one that helped with clot retraction. That wasn't on the test, but it was in your lecture notes. Okay. Actomyosin or actinomycin? Actomyosin. I think it's fine. Okay, 3A. Name the leukocyte in the field of view. 3B. Give me its percentage. Okay, and then we're going to move and look.
What? Was it eosinophil?
4A.
No, it wasn't basophil.
Tell me the leukocyte
That one's basophil.
and give me its percentage. We'll go backwards. What was for A and B? This is a what? Lymphocyte. It's quite small. You see, this should be even easier because this is a small size lymphocyte. They're only a little bigger than a red blood cell. And what? The nucleus takes up almost the entire field of view for the cell. It is a what? A granulocyte, but you can't see much of the A granularness because the nucleus is so large. Basophil would be a good guess, but it would be wrong because you would see clear, distinct blue grains outside of the nucleus. Okay? And what is the lymphocyte percentage? 20 to 25%. Some books even say as high as 45%. Now, if you said basophil less than 1%, would you get some credit? Yeah, you would miss basophil and then you would get less than 1%. So don't leave things blank on your test, because even if you miss part A, occasionally you can get a part B correct. And then, we'll come back to that one. And this is a what? This was 3A and 3B, neutrophil, and its relative abundance is 60 to 70 is fine.
Quick question, if we put like the minimum and a maximum percentage, is that fine? Like if somebody put 60% or 70%?
As long as it's within the range, yes, that's okay. All right, let's go to 5A and B. Tell me this large leukocyte, and if you have 20% of these in a differential count, what might that indicate? So that's 5A and B. Close your eyes. Okay, and then, name this, what are we on, 6A? Name this red blood cell disorder. And 6B, without seeing it, what was the white blood cell disorder that is a common type of leukocytosis? Okay, so 5A, what was it? Oops. This was a what? Monocyte. See the C-shaped nucleus? I gave you a hint, what? Large. And if it's 20%, that's chronic infection, yes. So what kind of blood test tells you about particular diagnosis? Differential count, all right? So that's why you need to know what the significance of the percentage of each different one is. Then 6A was this. What's that? Red blood cell disorder, sickle cell anemia. And 6B, what disorder did you see that was a common form of leukocytosis? Leukemia. So if you had a leukemia slide, the clear
What?
feature is what? Many white blood cells in the field of view. Okay? So we'll pause there with that, and that takes you through a lot of the microscope part of the test. So what have we done? About 12 questions on the formed elements and the particular leukocytes, all of that. Are we done with chapter 26 yet? No. There's now the blood tests. We integrated one blood test into our questioning. What was that? Differential count question. Then, 7A, define hematocrit. 7B, abnormally low hematocrit might indicate what? 8A, tell me one of the two blood tests for hemoglobin concentration. That's 8A. And 8B, abnormally low numbers for this test might indicate what? 8A was tell me one of the two tests for hemoglobin concentration, and 8B was tell me what an abnormally low number for that test, what it might indicate. Okay, are you ready? So 7A, define hematocrit, percent of red blood cells, right? Or red blood cell volume, packed cell volume, you could say that. 7B, anemia. Abnormally low hematocrit might indicate anemia. Or what if I gave you a number? For a female, hematocrit of 55 might indicate what? Blood doping or polycythemia, sure. And hematocrit hematocrit of 32 might indicate what? Anemia. So you should know the relative percentages for hematocrit also. So that was 7A and B. 8A was what? What's that? Yes, name one of the two ways that you test for hemoglobin. One is called the Tallquist method. That was little sheets of white paper that you compare the color of red against a scale of kind of orange to deep red. Or there's a little machine, you put a drop of blood in the machine, look at it up to the light, and that's called a hemoglobinometer. So either of those would be fine for indicating blood tests. What other blood test did we do? Well, what blood test would we use an unheparinized tube in? And we're looking for a time for normal being 2 to 6 minutes. That's 9A. So which test would we use an unheparinized tube in it? And we're looking for a time for this event to occur in about 2 to 6 minutes. That's 9A. 9B. What antigens are found on O negative blood? A. Mismatching ABO blood types would cause agglutination or coagulation. Choose one. That's 10A. Mismatching blood types. What's that? For 10A? 9A. What did I say for 9A? Oh, yeah.
He just said mismatching.
I have it on video. So I can always go back and look at it.
Anemia again. Abnormally low value for Tallquist would be anemia again. That was what?
What's anemia? 8B.
That was B for what? 8B. Okay. And then we need a 10B question. 10B. Tell me, what is the pH of blood? Okay? So 9A. What was the test that we would use the unheparinized tube in? Coagulation time test. And 9B. What was the question? I forget. Oh, what antigens are on O negative blood? None. (10A). What happens if you mismatch ABO blood conceivably? Agglutination or coagulation? Agglutination. And the pH of blood is 7.35 to 7.45.
We have to know the 0.35 to.
Yeah, because it's a 1/10 of a unit's range, pretty tight. Okay, 11A. Is that what we're up to? 11A. How about what would happen if you gave A negative blood to someone who's AB positive? Choices are coagulation, agglutination, or nothing? (11B). In a hematocrit test, what is the upper portion of the tube full of? What component of blood would you see in the top of the hematocrit test? (42), what do you call the little white line underneath of that clear upper portion in the hematocrit test? And here, we'll do those two and then we'll switch to heart questions. Okay, so what was 11A? If you gave A negative to AB positive, what would happen? Agglutination, coagulation, or nothing? Nothing. Right? There's nothing different in A negative than what AB positive already has. Then the next question was, in the hematocrit test, what's the top portion of the volume? The plasma. What's the white line? Bonus 42? The Buffy coat. Okay, so that's about a week's worth of lab, right? So that was your whole blood lab. We just did 22 questions. I think I said maybe 20 to 24, right? So do you agree from your review? Did we cover these things? Identify the components of centrifuge blood. What would that be? Basically your hematocrit. What are the components? Can you tell me the physical properties? Yeah, I asked you what? pH. Could be a different question. Then, microscope. Lots of what? Identify and give functions of all the formed elements, including the different leukocytes and their percentages. So that's basically knowing the platelets, the red blood cells, and all the 5 different leukocytes, and the percentages integrates in what? The differential count. Agree? Disorders, leukemia, sickle cell anemia, and then don't forget to look over your blood tests, because we had to describe